Hi, class, and welcome to Chapter 9, where we'll be talking about the patterns of inheritance. We'll briefly introduce genetics. We'll do a little introduction here. I've also included a YouTube video to watch that kind of helps explain a little bit more of Mendel's genetics and Punnett squares. So take a look at that YouTube video found under this chapter in the modules as well. So as of 2018, over 12 million Americans have sought to learn about their genetic makeup, so the genes. Within weeks, they receive a report providing a large amount of genetic information, and the surging popularity of these services proves how deeply many of us care about our genetic makeup. Our genetic makeup is important because our genetic makeup can tell if you're predisposed to a disease, what your kids might look like, and things like that. So the big ideas that we'll talk about in this very long PowerPoint are Mendel's laws, variations, the chromosomal basis of inheritance, and then the sex chromosomes and sex-linked genes. I'll be covering a few of these in this lecture PowerPoint. I won't be lecturing on everything because I don't want to bore you, so I include some YouTube videos interspersed as well. So first of all, Mendel's laws. We'll talk about Hippocrates first. He had the idea that pangenes travel from each part of an organism's body to the eggs or sperm. And this is incorrect. It was an incorrect hypothesis because your reproductive cells are not composed of particles from somatic cells, and changes in somatic cells do not influence your gametes. This blending hypothesis was rejected because it did not explain how traits that disappear in one generation can reappear in a later generation. So this was Hippocrates with kind of his early genetics ideas. Then we get to the science of genetics and how it began in an abbey garden with Mendel. Hereditary is the transmission of traits from one generation to the next, and genetics is the scientific study of hereditary, and it began with a man named Gregor Mendel and his experiments. He found that a heritable feature that varies among individuals is called a character, and Mendel crossed and planted pea plants and traced all their traits, which are just variants of a character from one generation to generation. He hypothesized that there are alternative versions of genes or alleles, and these are the units that determine a heritable trait. So here's a look at Gregor Mendel. He lived from 1822 to 1884, and he used pea plants. So here's the anatomy of a garden pea flower with one petal removed to improve the visibility. And what he did is Mendel removed the stamens from a purple parent flower, and he transferred the pollen from the stamens of a white parent flower to the carpel of a purple parent flower. So he's basically doing a fertilization experiment between purple and white parent flowers. When he did that, the pollinated carpel matured into a pod. So he basically is growing a new pea plant. And from that, he's able to plant the seeds from the pod and observe the traits of the offspring to tell which traits between parents are kind of seen in the daughter. So the long description is what he did is the 7 pea traits studied by Mendel during his cross fertilization were the character, the dominant trait, recessive trait, the flower color, the flower position, the seed color, the seed shape, the pod color, and then the stem length. So he found dominant traits and recessive traits, and he was able to kind of list all these traits to see what the daughter plants would have and which ones would be kind of seen in the daughter plants. So he was looking at the daughter characteristics of what their flower colors were, their flower position, their seed colors, the seed shape, the pod shape, pod color, and stem length. So again, Mendel crossed and bred plants together and then tried to figure out how what the probability was to get these different traits. Mendel, from all of his experiments, developed 4 hypotheses, and they're described below using modern terminology. There are alternative versions of genes called alleles that account for variations in inherited characteristics. And for each character, an organism inherits 2 alleles of a gene, one from each parent. An organism that has two identical alleles for a gene is said to be homozygous, and an organism that has two different alleles for a gene is said to be heterozygous. If the two alleles of an inherited pair differ, then one determines the organism's appearance and is called the dominant allele, and the other has no noticeable effect on the organism's appearance and is called the recessive allele. And a sperm or egg carries only one allele for each inherited character because allele pairs separate from each other during the production of gametes, and this statement is called the law of segregation. So here's a cross that tracks one character, the flower color. The F1 generation, all plants have the purple flowers. And then if we cross that again, we'll see that one of the daughter plants will have a white flower. So we get 3/4 of plants have a purple flower, but 1/4 of plants will have a white flower in the second generation. Mendel's hypothesis also explains this 3-to-1 ratio observed in the F2 generation. So the F1 hybrids all have the PP genotype. A Punnett square shows the 4 possible combinations of alleles that could occur when these gametes combine. And this again just shows how the lowercase p stands for the recessive trait, being white, and the uppercase p stands for that's the dominant trait. So we get this Punnett square where we separate out the traits, where the uppercase PP are the dominant traits and the lowercase pp are the recessive traits. This idea of a Punnett square and how to create a Punnett square is described more in the YouTube video that I show as well. Okay, so every diploid cell has pairs of homologous chromosomes, and the chromosomes in a homologous pair will carry alleles of the same genes at the same locations. So this looks at 3 gene loci or locations on a homologous chromosome and just showing that the genotype PP, it's homozygous for the dominant allele, meaning they're both same for the dominant uppercase allele. Lowercase a, meaning it's homozygous for the recessive allele because lowercase always has to do with the recessive. And uppercase b, lowercase b, mean it's heterozygous with one dominant and one recessive allele. So these are just different locations on the chromosome for different genes producing a trait. The law of independent assortment is revealed by tracking 2 characteristics at once, and a cross between 2 individuals that are heterozygous for one character is called a monohybrid cross. A dihybrid cross is a cross between 2 organisms that are each heterozygous for 2 characters being followed. And Mendel's law of independent assortment states that the alleles of a pair segregate independently of other allele pairs during gamete formation. So that just means each allele is completely separate or independent of each other. So this just shows how the different alleles, capital R, capital Y, when they cross with a different type of pea flower will give you this kind of generation and characteristics. We get into a lot of Punnett square, and I won't ask you guys too many details of this. This is an example of a phenotype, a black coat, normal vision. That is the genotype. And when the offspring is mated with blind dogs, chocolate coats, or chocolate coat blind dogs, and what's the ratio of dogs that we would get? We would get a 9:3 to 3:1 ratio of Labradors for that. So here are 4 possible phenotypes and genotypes for coat color and vision in Labrador dogs when they're crossed. And we always get the ratio in a 9:3 to 3:1 ratio for crossing. Okay, so I will let you guys kind of read through and watch that YouTube video. This gives you a little more details on the Punnett squares. Let's talk about pedigrees and family trees though. The inheritance of many human traits follows Mendel's laws, and family pedigrees or a family tree can help determine an individual genotype. So for example, if we have someone with a straight hairline versus a widow's peak, which is kind of the triangular end at the top of the widow peak, we're able to kind of track through a family pedigree what the offspring might be. So a family pedigree map traits across 3 generations. Uppercase H alleles represent the widow's peak allele, whereas a lowercase h allele represents the straight hair allele. First generation grandparents are as follows. So we have Al and Beth are mating partners. Al is a male with an uppercase H and a lowercase h, widow's peak hairline trait. And Beth, a female, has an uppercase H, lowercase h, widow's peak hairline trait. If Charles mates with Debbie, Charles is a male with a lowercase h and lowercase h, straight hairline trait. Debbie is a female with an uppercase H, lowercase h, widow's peak hairline trait. The second generation parents, aunts, and uncles who are children that result from mating in the first generation. So you're able to see squares are males and circles are females. And if it's filled in, it means it has the straight hairline trait. And if it's not filled in, it's the widow's peak hairline trait. And you're able to see the traits as they're passed down through generations. So a pedigree can show the different traits that a family might have. So it's kind of fun to look at different pedigrees. The genetic disorders listed in the next table are known to be inherited as dominant or recessive traits controlled by a single gene. And most people who have recessive disorders are born to normal parents who are both heterozygotes, that is, parents who are carriers of the recessive allele for the disorder, but are phenotypically normal. So you can see these recessive disorders and the dominant disorders and what the major symptoms are. And I want you guys to know these because you might see them in your future healthcare related fields. So here's a look at an example of a single gene inherited trait in humans, such as freckles or no freckles. So freckles is actually a dominant trait. Recessive trait is no freckles. Even though it's a dominant trait, it's less common to have freckles. Pigment versus albinism. Albinism is much less common, and that is the recessive trait. So there are the freckles, the no freckles, the pigment versus the albinism. And this just shows, again, how the offspring produced by parents who are both carriers for albinism, which is a recessive disorder. Here's a look at a dwarfism. This is Dr. Michael Ain, a specialist in the repair of bone defects caused by achondroplasia and related disorders, and he has the dwarfism. Here's a look at fetal testing, how they can test a fetus for genetic disorders. And you can get a karyotype to show all the disorders of the phenotype. And this is karyotyping, which shows all the chromosomes, and it could possibly show any sort of genetic disorder. Traditional ultrasound scanning for a fetus. Okay, variations on Mendel's laws. Mendel's laws are valid for all sexually reproducing species, but the genotype often does not dictate phenotype in the simple way Mendel's laws describe. So Mendel's P crosses always looked for one of the two parental varieties, a situation called complete dominance. And for some characters, the appearance of the generation 1 hybrids falls between the phenotypes of the two parental varieties, and this is called incomplete dominance. Incomplete dominance, for example, in this snapdragon flower color will result in a pink flower, so not red, not white, but a pink flower, which is kind of a combination of the two. We call that incomplete dominance. Okay, incomplete dominance can also occur with cell diseases where we could get a mild disease or a severe disease or a normal disease. So with this mild disease of the example is a kind of between the severe and normal disease. This also has to do with the ABO blood group phenotype in humans that's controlled by 3 alleles that produce a total of 4 phenotypes.